Hi, my name is Rob Edwards. I'm the director of the University of California, Irvine Laser Capture Microscopy Facility. I'm going to be describing for you the use of the laser capture microscope that we have here. Our instrument is a Leica ASLMD, which we purchased approximately 3 years ago. There are two different types of laser capture systems. One can be likened to a spot welder, where one has a piece of tissue on a glass slide on top of a plastic membrane which overlies the tissue. A laser source is then used to melt the membrane, in effect spot welding the tissue onto the membrane, and then that membrane is removed, taking the captured tissue with it. Okay, to start the LCM system, we power on a number of instruments in sequence here. We'll start with the ASLMD control box, which is powered on here. We then turn on the computer. Simple enough. If you have a specimen that requires fluorescence optics for identifying the cells to be captured, you would turn on the mercury lamp for the fluorescence illumination. We're not going to be using that for this demonstration. If you want to capture high-quality images of the before and after specimens, one can turn on the spot camera separately right here. After the logic box and the computer are turned on, one next turns on the laser power control unit here, which is actually turned on by a key back here.